On. I'd like to introduce our speaker for today, Dr. Jolene Sutton from the University of Hawaii at Hilo. And without further ado, Jolene, take it away.
Hi, thanks, JC. Can you hear me okay?
Yes, I can hear you just fine.
Okay, perfect. Thanks for the invitation to do this today. My talk is about options for controlling mosquito-borne diseases in Hawaii. And to get started, I just want to say that this is basically a slightly more fleshed out version of a presentation that I gave at the Hawaii Conservation Conference in July. And so my co-authors are Cynthia King at DLNR and Floyd Reed at UH Manoa. And so a lot of the specific research details that I'll be focusing on a little bit later in the presentation, I just want to acknowledge Cynthia and Floyd. I also want to acknowledge up front that, the specific research that we're doing here in Hawaii, of course, there's a lot of people involved. And so at UHH, we've got a lot of students and technicians, and same at UHM. And we've also had a lot of really good discussions, especially with, Dennis LaPointe and Carter Atkinson. If you're talking about mosquitoes in Hawaii, then you're definitely in contact with Dennis and Carter. We've also been in communication with Scott O'Neill of the Eliminate Dengue program at Monash University in Australia. And a little bit more recently, we've been talking to people at Mosquito Mate and University of Kentucky as well. So I've indicated the funders on this slide. Again, this is sort of specific to the research that I'll get into later on. So I'm not used to giving presentations like this where I'm not in a room with people. So maybe, JC, before I go any further, how do you normally do questions for these kinds of talks? Do people interrupt and ask questions, or are they saved to the end?
That's 100% up to you. If you want people to shout them out as they have them, or wait until the end.
Okay. I'm pretty used to informal talks, so if anyone has a question during, I think maybe just please feel free to interrupt me. Okay. With that, I'll get going. So, mosquitoes, as we are all pretty aware, I'm sure, mosquito pathogens that cause disease. So, especially here in Hawaii, we're very familiar with recent outbreaks of dengue fever, scares about Zika, but they also vector a lot of other nasty things as well, like yellow fever, malaria, West Nile virus, chikungunya, and several others. Here in Hawaii, we've got 6 species of biting mosquitoes. So there's actually a few more species, but these are the ones that on this slide that actually do bite people and animals, and so these are the species that vector diseases. Collectively, these 6 species vector some pretty nasty things. So they vector things like Zika and dengue and West Nile. Some of these things already have been in Hawaii, but not all of them have, although certainly later on in this presentation, I'll be talking a little bit more about avian malaria and avian pox. So we've got these 6 biting mosquitoes here in Hawaii, and what many of you probably are already aware of is that none of them are native. None of our species of mosquitoes are native here in Hawaii, and so none of the diseases that they vector are native in Hawaii. If you go back through some of the literature I've got on this slide, this is just a screenshot of probably one of the first publications talking about mosquitoes in Hawaii. So this is a publication from 1904, and it's a bulletin from the Hawaii Agricultural Experiment Field Station. And so this is probably the first record describing how mosquitoes were first introduced to Hawaii. And so it's believed that the first species, the southern house mosquito, was introduced in 1826. Basically what happened was a ship, the HMS Wellington, was coming up from Mexico, and when it reached Lahaina, Maui, people on the ship replaced they got fresh drinking water, and when they did that, they dumped their old water into the streams. And so it's believed that that was the first introduction of mosquitoes in Hawaii, around 1826. And it's really interesting. I've included a link to this publication here at the bottom of the slide. It's really interesting to go back through some of these older references about mosquitoes, and it's interesting because they include the steps that were taken to first describe how mosquitoes were discovered in Hawaii, but also the early control efforts for mosquitoes. And so in that document that I've provided the link for, this is just some of the images. So they've included images of typical breeding habitat for the mosquitoes. What you can see, I know these photos are a little bit grainy, black and white, not the clearest, but I wanted to include the actual photos from that first publication. You can see that we've got images of basically water containers near human habitat. And on the next slide here, we've also got, so this is at that agriculture field station in the document, where they're trying to fill in the breeding habit for the mosquitoes. And so very early on, people were interested in controlling mosquitoes in Hawaii. If you go back and you look through these older documents, it's really interesting to start to put a timeline together for the introductions of mosquitoes in Hawaii, then the introductions of the diseases that the mosquitoes vector, and then also a timeline of control efforts. So I've got on this slide just sort of a breakdown of the early years. So on the left-hand side, 1826 roughly is when that first species was introduced here in Hawaii, Culex, or the southern house mosquito. Not too much longer after that, it's believed that Aedes aegypti was introduced, and so Aedes aegypti is well known as being a very competent vector of dengue, for example. And then in 1900, the first vector control program was established in Hawaii. Now this first vector control program was, if I'm correct, so I have to double check, but I think it was mostly targeted at rodents at the time. But getting that idea of controlling disease vectors was definitely part of those early control programs, early years, shortly after mosquitoes were introduced. And then a little while after that, Aedes albopictus. So Aedes albopictus is the Asian tiger mosquito. It's probably one of the most common mosquitoes here on the islands. And these three are also the ones that are typically most responsible for vectoring the things that we worry about here in Hawaii. And so speaking of the things that these mosquitoes vector, it was about 1902, 1903 that people started noticing in our bird populations evidence of avian pox. And so at this time, the distribution of avian pox in birds on the landscape was used to sort of assess the fact that Culex has become well established across the landscape. 1902 and 1903 was also the first dengue epidemic here in Hawaii. And this is really interesting because, of course, the most recent dengue sort of epidemic and scares in Hawaii had, I think maybe 150, 200 cases. It was a relatively low number by comparison to this first epidemic, which was 30,000 cases. So that was pretty phenomenal. And it was in 1904 that the first formation of the Hawaii Citizens Mosquito Campaign Committee was formed in Honolulu. And so what's interesting is that I'm here today talking about mosquito control in Hawaii, but we've really been talking about mosquito control in Hawaii since at least 1904, and we're still working on this problem. Okay, so the earlier slide that I showed with the image of the person back in 1904 filling in the breeding habitat for mosquitoes. So the breeding habitat is, of course, critically important for vector control. And the way that mosquitoes breed, you're probably familiar, is that after mating and after the eggs have developed, the female lays her eggs in water and the larvae hatch out into the water. They develop through several instars, then they pupate, and then they eclose to become adults. What's missing, however, from this image, as I'm sure you're aware, is the breeding. So once a female has mated with the male, she needs a blood meal. She needs a protein source to be able to develop her eggs, essentially. So most people, funny, aren't actually aware, I find in Hawaii, but it's only female mosquitoes that bite, so males never bite. And then the way that disease transmission happens, and again, you're probably mostly familiar with this, is that we've got a primary vertebrate host. And in this case, I've included an image of a bird, but this could be a person, this could be other types of animals as well. And so if a mosquito bites a bird that has a pathogen that the mosquito is able to vector, well, that mosquito becomes infected with the pathogen, and then it bites the next individual and passes that pathogen on, and then another mosquito comes along and bites that infected vertebrate host, and so the cycle basically continues. Since the more recent outbreaks of dengue in Hawaii, the Spite the Bite program has become really, really good at making people aware of how mosquitoes vector diseases and the steps that we can all take to try to mitigate the spread of mosquito-borne illnesses. And so you've probably seen this kind of flyer with all of these suggestions for helping to eliminate mosquitoes, and these are great. These have done a lot over the, since 1826 in the history of mosquitoes in Hawaii. Doing these kinds of steps, basically reducing mosquito habitat around homes and populated areas, has done a lot to help fight diseases such as dengue and things that are vectored by primarily Aedes aegypti, which tends to have really close associations with humans. So it's done a really, really good job. But the species that I want to focus on for today is the southern house mosquito. And so this is the species that I primarily work with in the lab, as well as Floyd Reed at Manoa. This is the species that I'm sure everyone is already aware of. It vectors avian malaria and avian pox. It's pretty widespread across the landscape. And so an effort to control the southern house mosquito and the diseases that it spreads really needs a larger scale effort than simply controlling mosquito habitat just around our houses. So Floyd and I have been working with Culex now in the lab for a few years. We established our first colonies in 2013 at Manoa. And at the time, we were working to genetically modify this species of mosquito. The overall objective or the long-term goal of this project is to develop genetic strategies for mitigating vector-borne diseases. And so just to kind of give you an idea of what we've been doing, I've got some images on this slide that show in 2015 we were able to genetically modify the species. All we've done in this case is sort of a proof of principle to show that the rearing techniques and the protocol that we were using to do modifications, that we were basically doing it right. So I've gotten this image in panel A. You can just see mosquito eggs lined up against some filter paper to get an idea of what they look like. Because it's the mosquito eggs that we work with when we're trying to genetically modify something. In panel B, there's two larvae. The larva on the top, it's fluorescing red. That's a genetically modified individual. So it's actually expressing a protein, DS red, that fluoresces under particular light. In panel C, it's the same two larvae, but they're shown under natural light. And in this case, it's the lower larva, the one with the yellow arrow pointed to it, that is the genetically modified individual. And you can see a slight difference in coloration, but not nearly as strongly as the difference in panel B when they're under the fluorescent light. In panel D, we've got two individuals. One is modified, they're pupae, and you can see that fluorescence in the lower individual, the one with the yellow arrow pointed at it. And in panel E, we have two adults. One is genetically modified. It's harder to see the fluorescence in the adults. It's very concentrated just in the thorax area, so the individual on the lower right is the fluorescing one. You can hardly even see the individual in the upper left. In the next panel, panel F, it's the same image as in panel E, except that the individuals are under natural light. And so you can see the differences there in the fluorescence of the DS red. So the reason that we're interested in Culex, specifically here in Hawaii, has to do with conservation, and particularly with conservation of Hawaiian honey creepers. And so, again, I'm sure everybody is in this at this talk present today is familiar with the story of honey creepers and the other avifauna here in Hawaii, and the amazing radiation of honey creepers that we have. And so the critical point to remember is that for Hawaii's birds, especially the honey creepers, which has been here for a very long time, they've evolved in the absence of mosquitoes. So for about 6 million years, these species have evolved completely isolated from mosquitoes, which means that they've been completely isolated over their evolutionary history for the last 6 million years from mosquito-borne diseases. So they've never had any selective pressure to be able to respond to mosquito-borne illnesses. And due to this isolation, not just due to mosquito-borne illnesses, but due to the isolation of these species, many of them are, extinct or endangered, critically endangered, or listed as vulnerable. And the thing to notice on this slide is the probable extinction dates. So starting back in the 1800s, we've got some dates recorded, but moving forward in time, going down that list of dates into 1985, 1989, 1988, or 1998 and 2004, we're still seeing extinctions. And again, not all of these extinctions and not all of these threatened species listings are due to mosquito-vectored illnesses, but a lot of them are, especially for the honey creepers. And so there was a link that was made back in 1986 between mosquito-vectored diseases, the distribution of mosquito vectors and the parasites that they vector, particular malaria, and the location of these native bird species. And so probably many people, again, are familiar with this image from the Van Riper paper in 1986, like I said, is that as you increased in elevation, essentially you would cross a threshold where native birds were able to better persist because they were not as vulnerable to the mosquito-vectored pathogens. Basically, there were sort of more optimal climates for either the mosquito vectors or the pathogens that they were vectoring, which was at lower elevations. And so you often hear talk in Hawaii about sort of the mosquito line or the mosquito disease line, and so we definitely see that in terms of our bird species being able to persist at high elevation refugia. And so to give you an idea, here's a map of the island of Hawaii, and we've got some high elevation sites that are basically the only refugia for many of these species. So Mauna Kea with Hakalau Forest Reserve is indicated in the map as well. At about 1,900 meters, that's about where sort of that safe line sort of starts to exist, around that 1,500 or 1,900 meter mark. And so these are the points on Hawaii Island that most of our birds persist in. Now, usually when I get to about this point in my presentation, or when I'm talking to people about these high elevation refugia sites, I get asked about Hawaiian amakihi, and I get asked about, won't these birds just develop resistance? And so my answer to that is there is an example of at least one species that is adapting to avian malaria, it seems, vector-borne diseases, which is Hawaii amakihi. And so if you look at this, this paper that was published in 2005, basically what was found in this paper is that low elevation populations Hawaii amikihi, those populations that were most exposed to, say, mosquitoes and the diseases that they vectored, these populations of birds were starting to come back. They were starting to rebound. And so this is great. This is excellent. This means that these populations had the genetic diversity in them, at least some of the populations, to be able to tolerate these diseases, and so the numbers are starting to come back. However, this is not what we see with every single species. We're very lucky with Hawaii amikihi, but it's not what we're seeing with every single species. We're still seeing species that we predict are facing extinction due to the mosquito vector diseases. And so the two species that get talked about the most in sort of more recently are two species of bird on Kauai, and Kauai forest birds in general, but especially the akikiki and the akeke'e. And the reason is that in the last, in recent years, these two species have started to crash. And of course, the Kauai forest bird recovery team would be able to tell you a lot more than myself, but it looks like these species are crashing and are predicted to go extinct if nothing is done in the very near future. And the interesting thing is that it seems to be linked to climate change and that sort of mosquito vector disease line. And basically, as temperatures warm, that line is getting higher. These populations on Kauai are right at that threshold of now being more exposed to mosquito vector diseases. And so this is an ongoing problem that we're dealing with here in Hawaii, despite having talked about control efforts since at least 1904. So that brings me up to the more recent times. So at the IUCN meeting, or shortly after the IUCN meeting that was held in Honolulu, a group of scientists and researchers and conservationists and Department of Health people all got together at KMC here in Volcano to talk about possibilities for mitigating mosquito-borne illnesses here in Hawaii. And the result was this white paper, which is available online, and it's called To Restore a Mosquito-Free Hawaii. But essentially the whole idea behind this meeting was just to get about 40 of us all in a room and just talking about different strategies, all kinds of different strategies. And so we talked about mosquito control methods, conventional methods, such as reducing habitat, reducing that breeding habitat. We also discussed traditional pesticides and biopesticides. And we also started to talk about more alternative techniques, sort of more recent techniques, I guess. And so some of these strategies, these alternative strategies, included sterile insect technique, Wolbachia, and of course genetic strategies. And I've highlighted the Wolbachia on this slide, and the reason I've done that is because the conclusion of this whole group of people from all over the place. We had people from the mainland, we had people from Canada, we had people from China and Australia. Everyone came away from this meeting feeling very excited about this Wolbachia technique. So this is the one that I'm going to focus the rest of my talk on, and it's this strategy that Floyd and I received funding from recently from DLNR and U.S. Fish and Wildlife to try to develop here in Hawaii Wolbachia strategies. So I'll walk you through what Wolbachia is and then what we're actually trying to do in our labs here at UH Manoa and UH Hilo. So Wolbachia is a bacteria, and it naturally occurs in about 70% of all insect species. So it's an endosymbiont. You can sort of think of it like the E. coli that we have naturally living in our guts. Wolbachia sort of has a similar relationship with insect hosts. And so this is an image of a cell, an insect cell, and it's infected with Wolbachia. And so I've just put some colored images on top of the cells to show you where the Wolbachia is. Wolbachia is really interesting, though, in that so it has some interesting effects on its insect hosts, in particular the insect host reproduction. And so to kind of give you an idea of where I'm going with this, basically Wolbachia uses its insect host reproduction so that it can help basically proliferate itself. And so it's evolved very, very close associations with its very specific insect host. And what that means is that if you have, different insect species, different insect cell lines, you very likely have different strains of Wolbachia because they've evolved so closely with their insect hosts. And like I said, the Wolbachia has very interesting effects on the insect host reproduction, and it does a few different things, but one of the things that it's most known for is this thing that's called cytoplasmic incompatibility. And cytoplasmic incompatibility, I'll walk you through what it does, but it's basically been used in other places as sort of a form of birth control for mosquitoes, and it's the factor or the type of control that we're looking at trying to establish in the labs here at UH. So the way cytoplasmic incompatibility works is that if you have a mosquito or any insect, basically, it has its particular type of Wolbachia. And so you take that individual and you mate it with an insect of the same species that has the same type of Wolbachia. Then they can mate and they'll have viable offspring. And those offspring will grow up and have the same type of Wolbachia as its parents. And actually it gets its Wolbachia from its mom. So Wolbachia is maternally inherited. On the other hand, though, if you take, say, a female mosquito with one strain of Wolbachia and you mate her to a male that has a different strain of Wolbachia, then the mating can produce eggs, but those eggs aren't viable, and so they don't hatch. And so what you end up with is basically what we've been sort of calling this birth control for mosquitoes. And what's really interesting is that this exact strategy has been used, like I said, in many, many places for very effective, very safe mosquito control, so mosquito disease control. And it was a very convenient time when I gave my talk at HCC because just that week there was a mosquito control using cytoplasmic incompatibility that was used in California. But this technique has been developed by the Eliminate Dengue program. They use Wolbachia. They actually use a slightly different strategy than cytoplasmic incompatibility, but they use Wolbachia to control dengue in places like Australia. Then there's Mosquito Mate, which is a mainland U.S. company, which have been developing techniques with cytoplasmic incompatibility. And most recently, Virally, which is associated with Google, has developed techniques to mass rear and deploy these mosquitoes. And so the efforts that I just mentioned a moment ago, this is a blog post that was published on the 17th of July this year, talking about how Wolbachia-infected mosquitoes, male mosquitoes, were being released in Fresno, California, basically to do exactly what I just showed on the previous slides, which is if you release a bunch of males that have a different type of Wolbachia than the females, the wild-type females on the landscape, those males mate with the females, but due to cytoplasmic incompatibility, because they have the different Wolbachia, you basically end up with that birth control. And so this was just done, like I said, this past year in California. So this is exactly what we're trying to work with and establish here in Hawaii. And from our perspective at UH, we're trying to work with just things that are already here in Hawaii. And so I'm going to walk you through the steps of what we've been up to. The very first thing is you need to get mosquitoes in your lab. So you need to establish mosquito colonies in the lab. And like I said, we started doing this back in 2013 at Manoa, and then more recently we started doing this as well in 2016 at Hilo. And so here's some images from our labs. These are Culex mosquitoes. We've got some egg rafts in the bottom left, and you can see that that's one egg raft from one female. And so one female Culex will lay anywhere from about 50 to 150 eggs per raft. They hatch out into those larvae. Then we've got the adult males and females. So in that image with the M and F over it, the male is on the left. He's kind of got that funny mustachy antenna. And then the female is on the right. She's a little bit larger and she doesn't have the fuzzy antenna. And then you can see an adult cage in the far right. And the adult cage that you see is 1 foot by 1 foot. And I mean, it's holding a few hundred mosquitoes in that one particular cage. You can see how we maintain them on sugar water, which is what's in that specimen cup. And we've just added a container to that cage to get eggs. And so we sort of have an eggs on demand system where if we're just maintaining the colonies, we put in the container of water, and the next day we'll collect many, many egg rafts in that container. And if we're trying to get eggs to do our micro-injection, either with Wolbachia or with genetic plasmids, then we time it so that we can collect eggs within about an hour of adding that oviposition water, that egg-laying water. So we have our mosquitoes. The next thing we need to do is clear Wolbachia, because Culex already have Wolbachia in them. And so we use an antibiotic to get rid of the Wolbachia that they already have. And the antibiotic we use is tetracycline. You've probably heard of tetracycline. It's used a lot in acne medication. It's also used a lot for livestock and on farms. In the image, the square image, what you see is you see a gel. And the gel has 8 lanes in it, 8 columns that are indicated, and then a 9th one on the very far right that's not labeled. But what we've done in this, for any of you who are familiar with running gels for DNA, basically what we've done is we've cleared Wolbachia from some of our colonies, and we've left some of our colonies alone, so we haven't treated them with antibiotics. And then we use PCR to test whether or not we've cleared the Wolbachia. And so we've tried to amplify a Wolbachia gene in each of these 8 samples, or 7 samples. So the first 5 lanes that you see, those are samples that have been treated with antibiotic. And so you don't see any bright bands at the top. So that indicates that we did not amplify the Wolbachia gene, so there's no Wolbachia in those samples. In lanes 6 and 7, you see those really bright bands at the top. That's because we were able to amplify the Wolbachia DNA in those mosquito samples, and that's our wild type one, so we did not treat those. And then lane 8 is just a negative control, and then lane 9 is just a DNA ladder for standard assessment. So we were able to use the antibiotics to clear the Wolbachia. It's basically the take-home message from this slide. Then we mask rear the Wolbachia-free Culex colonies, and so that means that we've got lots and lots of cages of wild-type Culex, and we've got lots and lots of cages of Wolbachia-free Culex. And it took a little bit of time to get the conditions established to be able to sort of rear our mosquitoes, but once you get things going, it's pretty easy to rear them. So what you're seeing in this image is the inside of a cage. You're looking into a cage, and there's a white tray, and that tray was put in the evening before, and it was left overnight. And all those dark spots that are floating on the water in that white tray that look like flax seeds, those are all Culex egg rafts. So there's about 80 or so egg rafts in that cage just from one night. So we've established Culex colonies. We've cleared them from Wolbachia, and we've mass reared those. The next thing that we need to do is we need to get a different type of Wolbachia than what Culex naturally has. And so we get that actually just from a different species of mosquito that is already here, although we're also working with other insect species as well. And so we're working with Aedes albopictus, which has a different type of Wolbachia than Culex does. We're also working with fruit flies, Drosophila melanogaster, which has a different type of Wolbachia as well. And to isolate the different strains, essentially what we're trying to do is basically collect those live mosquitoes fresh, crush them up, and sort of filter them so that we can get the cytoplasm from the cells, and the cells contain the Wolbachia, or the cytoplasm then will contain the live Wolbachia. And we try to inject those live Wolbachia into eggs, right away so that they'll survive. And so here's the setup for our infecting our Wolbachia-free Culex with this new Wolbachia strain that we're getting from Aedes that we've collected here in Hawaii as well. And it's a little difficult to see from this slide, so I'm just going to skip to the next slide. Basically, the way it's done is after we've crushed up those other insect species and we've filtered the cytoplasm, we use a very, very fine glass pipette to inject it into those eggs. And so this image is a little bit funny, but what it's meant to show you is I've got a glass slide. We've got some eggs lined up along filter paper to sort of brace them, which is very similar to a photograph that I showed earlier. And then we manually, are injecting each one of those eggs with a very fine microscopic needle with that slurry. And so, for instance, in the photograph on this slide, I've got the number 33 written down, and that means that there's 33 eggs that we're injecting on that particular slide. So we keep track of the numbers that we inject, and then out of those injections, the numbers that survive, and then out of the ones that survive, the numbers of females that hopefully are infected with the different strain of Wolbachia. So the stage that we are at right now is at that trying to infect the Wolbachia-free Culex with the new Wolbachia strain. And we are also screening the ones that we have injected, and then the next step would be to test for cytoplasmic incompatibility. Other studies have shown that the Wolbachia from Aedes albopictus does cause cytoplasmic incompatibility when it's introduced into Culex, but of course we would have to confirm that in our lab as well. The other stage that we're at is the community engagement stage. This is where we're a little bit behind, and so I'm really grateful to be able to talk to everyone on the phone today, is because we really want to get the word out there about what we've been doing. And so I was at a Science Cafe event last week in Honolulu and doing this today, and basically we just need to start talking, and we need to find out from the general community. We need to educate them and also find out how they feel about these different types of possible control strategies for mosquitoes. What we're trying to do at UH is to put a tool on the table so that it can be available at some point, but it's only going to get used if, of course, the public accepts it. And so I've got an image here from an Eliminate Dengue kiosk, because I really consider these guys the model for community engagement. They have done a lot of work using Wolbachia-infected mosquitoes, like I said, in different places of the world, but especially in urban areas in Australia. And coinciding with their with the work and the research that they did, they put in a lot of effort to survey the community and do a lot of community outreach events. And they've published the results of those surveys. and also what they have done to address the public concerns that came up in those surveys. You know, whether this technique is safe, can it spread to people, to anything else. They actually addressed a lot of those concerns already in their public engagement and in their surveys and the publications, like I said. So I really consider these the model group that we you know, are trying to now follow. But that's really it for me today. So I just want to say thanks to everyone for listening. If you have any questions, I'm happy to take them.